Last time we built a virtual heart inside a computer, but no matter how precise the model, it needs something real to validate it. CiPA's third pillar is a real, self-beating heart cell, and its origin is almost magical. Take a single skin cell from your arm, and you can turn it into heart muscle that beats, thump after thump, in a dish. You can even build a specific patient's own heart and replay their disease in that dish. This is thanks to a Nobel-winning technique, reprogramming. Scientists insert a few specific factors into an ordinary skin or blood cell and rewind it into a stem cell that can become anything, an induced pluripotent stem cell, iPSC. Then, given the right signals, it differentiates step by step, and within weeks, a sheet of self-beating human heart muscle grows in the dish. Why is it indispensable? Recall the first two pillars. The in vitro panel takes the channels apart and measures them one by one. In silico reassembles them inside a computer. iPSC heart cells are a whole, living, complete human cardiomyocyte, where every ion channel naturally sits together, works in true proportion, and is human with no dog or rabbit species gap. It supplies what the first two pillars lack most: reality and wholeness. So how do you read it? The workhorse tool is the microelectrode array, MEA. Spread the heart cells over a chip covered in tiny electrodes, and each electrode records the cell's field potential, essentially a miniature ECG. Add the test drug, and you directly see whether this cell's repolarization drags out and whether abnormal beats appear. You can also use voltage-sensitive fluorescent dyes to watch the action potential with light. How accurate is it? CiPA ran a blinded test, 28 drugs of known risk, their names hidden, handed to iPSC heart cells across different labs. The predictive accuracy for arrhythmia risk reached about 87%. Even better, if the cells come from a patient with an inherited arrhythmia, they replay that person's disease, say long QT, right there in the dish. That makes it not just a drug test, but a tool for disease modeling and even precision medicine. But let's be honest, it isn't perfect. Today's iPSC heart cells are still immature, more like a fetal heart than an adult one, with a resting potential that sits high and unsteady, exactly because of the scarce IK1 we covered in episode 6. They also beat on their own, and their channel expression and calcium handling differ from adult muscle. That immaturity sometimes discounts the results. How to mature them is the field's most important challenge right now. Put the three pillars together, and you see the whole picture of modern cardiac safety. The in vitro panel measures how much each channel is blocked. The in silico virtual heart folds that data into a risk score, and the iPSC live cell cross-checks it with a real heart. Only when all three corroborate can you truly say whether a new drug clears the cardiac safety gate. And with that, this season has traveled from a single tiny hERG channel all the way to a precise system called CiPA, and the foundation beneath every step of it has always been patch clamp. Next time, we cover how to turn this whole body of evidence into a report regulators actually accept. ICHE14, S7B, and the thorough QT study. Follow Channel Observatory, and we'll take them one channel at a time.